We talked some about the use of antibodies with respect to measurement of hormones. I also mentioned the fact that antibodies, labeled antibodies, can be used for localization as well as quantification of hormones. I want to talk some, or give you some background information that applies to both chemical and biochemical, immunological, even bioassays. It applies to all analytical assays. There are 4 parameters that govern the performance of any of these assays. One is specificity, and that's the uniqueness of the detected species, the lack of cross-reactivity. That means there's not a metabolite or denatured molecule or a stereoisomer that can interact with your binding agent or your detector, so that what you're measuring is actually what you want to measure. The parallelism of diluted samples is a refinement we can talk about in more detail if we've got time. But the main thing is that it's the uniqueness of the detected species so you don't get cross-reaction with other things that are present. Precision is the reproducibility of measurements. Recognize that if your assay is nonspecific, you may have a very precise assay. You can reproduce it. But the measurements you generate may mean nothing. You may be precisely wrong. Okay. So these two are different parameters. Accuracy refers to the ability to estimate the correct or known amount of an analyte contained in standards or reference samples. One of the things that you end up doing when you're developing assays, particularly immunoassays, is you have to at some point cross-check them against samples. Let's say you're dealing with steroids. With steroid samples that have somewhere been measured by another means, say mass spectrometry, where you know that you're dealing with a pure material that you can weigh out on a scale, that you can dilute down in a serial dilution and use as your standards in your immunoassay. If you're finding that you're estimating the correct amount under those circumstances, then you have an accurate assay. You can get assays that are inaccurate because they allow for some sort of cross-reactivity between the binder and the ligand, or between the ligand and something else in the sample. And under those circumstances, you will often introduce a bias in the assay. So there's, you can get comparability, your comparability of the assays with the same analyte used elsewhere or in other assays. If you don't see an up or down movement from that, you have a lack of bias. If you do see an up or down movement relative to other assays of the same type or done in other labs, then you have some sort of a bias, which is a violation of accuracy. And then finally, you've got sensitivity. So specificity, the uniquenesses of response. Precision is reproducibility of response. Accuracy is correctness of response. Sensitivity is defined differently if you're a chemist or if you are a clinician. analytically, a chemist or biochemist would look at the slope of the assay response curve. How far up does the response go for every increment in added ligand or analyte? So the slope of the assay response curve, or the height in response to added analyte. Diagnostically, what you're looking at is the limit of detection, or the lowest measurable non-zero amount of the analyte. A diagnostic lab will actually look at both of these parameters, the slope of the curve and the ability to see the bottom of the curve, or see the low values. Let me give you a little picture that hopefully conveys that. Let's make this a hormone assay. Here's a response or signal. And our samples look like this. If I were to draw a best fit line through these, they'd go up something like this. If I were to draw error bars at each one of these, they'd look like this. Right? If those error bars are drawn out to 2 standard deviations, or sometimes even 3, you get an envelope or an area on the curve that looks something like this between the dotted line, or between the dashed lines.
That's if what?
If these standard or variability that's shown is 2 or 3 times the standard deviation. Okay, what you get is an area of possible response for samples in the assay. So it's not a perfect line. It's not just one line. It's actually an area between these probability curves, this envelope. So an unknown, if you get an unknown out here, and you extrapolate it to the standard curve, it may be anywhere between there and there. So if you project that down, you get something called the error of the estimate. So it's not an absolute point. It's a window. All right. The steeper this slope is, holding the precision constant, the better you're able to distinguish one concentration from another, because this window becomes narrower as this slope goes up. Okay? But what happens down or at the ends of the curve is you've got this situation where this error of the estimate, if you've got a sample, let's say, measuring here, the the error of the estimate goes way back here out to here. So error of estimate includes 0. At the top end of the curve, you got the other problem where if you've got a sample that measures up here and you extrapolate it to the curve, you've got error of the estimate includes, what's up at that top end there? The ceiling, what would you call it numerically? Infinity. All right. So it blows up at both ends of the curve. Now, to be sure that the assay that or the results you're getting are actually readable and can be determined to be non-zero, you have to be above this window. So your sample has to be, say, somewhere here so that this error of the estimate is above 0. So the limit of detection is the sample with the lowest concentration that can be reliably estimated as above 0. Okay? Now you're all ready to walk into a diagnostic lab and to begin to apply that stuff. So, running a simple assay is not that simple. There are a variety of parameters that you have to fulfill, and these are the 4. Specificity, precision, accuracy, sensitivity. And it doesn't matter whether you're looking at slides and counting specks that are stained, or you're doing a biochemical assay using an enzyme, or you're doing a mass spectral analysis using a $1 million machine. They all have to follow those parameters. Before I get going too far, what's the time? Okay. All right, we burned another hour together. The next time I'm going to get into, I want to go through hypothalamic hormones and the control loops. And once we're through that, we can get into oncogenes and the interaction with the cancer system. Okay. Thank you. Yeah.